Hey guys, Miss King here, and in this screencast, we're going to look at transport. In our last screencast, we looked at the structure of the cell membrane. Now we're going to look at how it's actually working. So there are two types of transport across the membrane to help the cell maintain homeostasis. The first type of transport is called passive transport. What that means is there's no energy being used, so things can just pass in and out of the cell membrane. And those things are going to move with the concentration gradient. So think about like a river. If you were in a boat floating down a river, if you're going with the current, then that's kind of the same thing as whether you're going with the concentration gradient. So those molecules are going to move in the direction that the other molecules will move. Now for active transport, the second type of transport, that means that energy has to be used. And the reason that it needs energy is because it's going against the concentration gradient. So think of that as sort of like if you're going against the current in the river, if you're floating in your boat, you're having to use a lot of energy to move upstream. So active transport requires energy. What we're going to focus on in this screencast is passive transport. And there are three different methods for cells to do passive transport. The first method is called diffusion. Simple diffusion just means that molecules are going to move from an area where there's a high concentration to an area where there's a low concentration until they reach equilibrium. So when they go from high to low concentration, they're going with the concentration gradient. That's going to look something kind of like this. Here's our cell membrane. You can see the lipid bilayer here. And we have some material, some particles on this side of the cell membrane, and then we don't have any particles on this side of the cell membrane. So what that means is here we have a high concentration, and down here we have a low concentration. So what's going to happen is those molecules are going to diffuse across the membrane. They're going to start moving in that direction. They're going with the concentration gradient. They're going from high to low concentration. Once they equal out, once there's the same number of molecules on this side as there is on this side, that's what we call equilibrium. It's equal on both sides. And those molecules, they're going to stay in motion. They're not frozen, so they're going to keep moving. So what they're going to do is they're going to keep crossing, but they're going to go back and forth in order to keep that state of equilibrium. So they'll just keep going across the membrane forever if they can. The second type of passive transport is called facilitated diffusion. When you facilitate something, you assist it. So this is sort of like an assisted type of diffusion. So we're going to use our special protein channels or our carrier proteins. And sometimes we said that molecules are just too big, or maybe they have a polar charge, so they can't just pass through the lipid bilayer. So this is where they're going to use those special protein channels to be able to pass in and out of the cell. Okay, so the only difference between regular diffusion and facilitated diffusion are these protein channels. Now, the third type of passive transport is called osmosis. Osmosis is a type of diffusion, but it's only dealing with water. The key word for osmosis is water. Okay, so sometimes when those molecules are too big to pass through our membrane, there aren't any protein channels that will work with those molecules. So what will happen is we'll change the concentration by moving water from one side of the membrane to the other. So instead of moving the materials, we'll move the water. once osmosis occurs, it will kind of look something like this. Okay, so think about this beaker as on this side of the beaker, let's say that this is Kool-Aid, and this Kool-Aid has too much sugar in it. Okay, so it's too sweet. On this side of the beaker, we'll say our Kool-Aid doesn't have enough sugar. So these little molecules of sugar, see how spread apart they are? There's just not enough sugar here, so it's not sweet at all. This membrane will not let the sugar pass through, so the sugar molecules can't move from high to low concentration. So what will happen is the water molecules will move so that it will equal out the concentration. So now what this is showing you is that our concentration is the same on both sides. So now it is equally sweet Kool-Aid, and it's just right. How does osmosis affect cells? Well, there's 3 ways. An isotonic type of osmosis, iso means same. What's going to happen here is our water and our solute concentrations in and out of the cell are going to be the same. Okay? This means that the molecules will go in and out of our cells at equilibrium. So the key word for isotonic is that the cell is at equilibrium. This is what our cells want. They want to be at equilibrium. They want the concentration on the inside of the cell to be the same as the outside of the cell. The second type of osmotic situation is called hypertonic. Hyper means over or above. And what this means is with our cells, there's more water on the inside of the cell than there is on the out. And what that's going to do to our cell, that water is going to leave the cell because it's still going to go from high to low concentration. And when it leaves the cell, it causes the cell to crenate or shrivel up, kind of like a raisin. And then our third type of osmotic situation is called hypotonic. Hypo means under or below. So basically what's happening here is there's more water on the outside of the cell than there is on the inside. So the water is going to go into the cell, and this time it's going to cause our cell to blow up like a balloon. Now, when the cells blow up like a balloon, when there's too much water in them, they will do what's called lyse or burst. So literally, they bust like if you put too much air in a balloon, it will pop. I remember these. Here's a trick for you. It's hard to tell hypertonic and hypotonic. So the way I remember this, hypotonic sounds a lot like high pop tonic. So the hypotonic solutions are the ones that are going to make the cells pop. The water goes in and they swell up and get real big until they pop. So that's how I remember which one is which. Now, the types of cells that are affected are not just human cells or animal cells, but plant cells can also do osmosis, and they do. They can swell up and pop, they can be at equilibrium, or they can shrivel up. And this is usually, the hypertonic is usually what happens when your flowers wilt and kind of hang over and they just look terrible. So that's a hypertonic situation. That means it doesn't have enough water. We're going to have some problems that we're going to need to be able to work. So if we are given a situation where we have certain concentrations in our environments versus certain concentrations in our cells, we'll need to be able to figure out if it is isotonic, hypertonic, or hypotonic. So if we look at this first example, this is the environment in the square, and then the circle is our cell. So let's label that. This is the environment that surrounds our cell, and then this is our cell. Okay, so the environment, we have 100% water. And then on the inside of our cell, it says we have 4% salt. When we're talking about percentages, it always needs to equal 100%. So if there's 4% salt, then that means that there's 96% water. 96% is less than 100%. So if we move our water molecules from high to low concentration, that means they're going to go in to this cell. When the water goes into the cell, that's what we called hypotonic. And we also said hypotonic, when the water goes in, it causes our cell to swell up and then eventually burst. So this cell is going to lyse in this environment. For our second cell, we have 85% water on the environment. And again, our same cell, we have 96% water. 96 is more than 85. So for this cell in this environment, the water is going to go out of the cell. Okay, high to low concentration. 96 is high, 85 is low, so the water is going to leave this cell. Now, when the water goes out, we called that hypertonic. And we said when the water goes out of the cell, it causes it to shrivel up and crenate. All right, and then this environment, we have 95% water on the environment, and we have 96% water inside our cell. Now 95 and 96 are pretty close to each other, so what we're probably going to see happen here is a little bit of both. We're going to have some go out and some go in. It's very close to what we called isotonic, and that's going to be what we call equilibrium. Okay, and we'll call it equilibrium if it's within 1% of being equal. Okay, so if we look at one more example, here's another one. This cell is 15% sugar, so that means that it's going to have 85% water. All right, so if we look at our environment, 90% is more than 85%. So the water is going to do what? That's right. You said go into the cell. And so if the water goes in, it's going to blow up like a balloon. And that was hypoptonic or hypotonic. Okay, so this cell's going to lyse. All right, right here we have 15% sugar. So again, that's 85% water. And then on the outside, we have 16% sugar. So that means we have 84% water. Be careful when you're looking at the percentages that you pay attention to what molecules they're giving you. Okay, so 84% water is about the same as 85, so we're probably going to see a little bit of both in and out. And that would be isotonic. Okay, so that cell is going to be at equilibrium. All right, and then our last cell, we have 27% sugar. So that's going to give us about 73% water. And then the inside of our cell again is 85% water. All right, 85 is more than 73, so the water is going to go out of our cell this time. Okay, and when the water goes out, it shrivels up, and that's our hypertonic solution. And that's called crenating. So this cell is going to crenate or shrivel. Okay, and that's how we work the osmosis problems. We will have some of these in class, so be ready to work some of those out.